This is Holy Cross Component 1 Mock, and this is Mock number 1. First question. All of this is not very applied at all, and it's just straight biological knowledge, so this should be pretty straightforward on respiration. The molecule shown in the diagram is a nucleotide known in biochemistry as intracellular energy currency, and name the molecule. So you should recognize the molecule as ATP, or adenosine triphosphate. Circle a purine base on the diagram. Well, on here we've got this thing here, then we've got this thing here, and then we've got this thing, sort of thing there. And you should recognize a purine as a double ring from when you did DNA and nucleotides. So I've put circles around everything now, but the circle that we want is this purine here. So that's the purine. We've got the sugar and we've got the phosphates. Name two products that are found when the molecule in the diagram has released energy in cells, and it wants two. So the ATP is going to make a DP, and it's going to be a phosphate. So, and phosphate. Or you might have put inorganic phosphate there. Or you might have written the iron name. Why might this energy currency be described as universal? Well, it occurs in all cells, in all organisms, all metabolic reactions, so used for all reactions in all cells, in all organisms. Then we've got a little diagram. This diagram here, we can see, what can we see? We can see a membrane. So we recognise a phospholipid bilayer. There's another membrane there, and there's some things in that membrane. And this is respiration. So respiration membranes. This must be the membranes in a mitochondrion. So if I was just to draw this, we've got the outer membrane, and then we've got an inner membrane, and this could be a close-up of, say, that area there. They're both membranes. So this area here showing the two membranes. Name the area A and B. A is between the two membranes, so it's the intermembrane space. So intermembrane space. B's down here, so B is inside this mitochondrion, so that's the matrix. NAD, there we are, is used to transport hydrogen ions, and there looks like a little wagon, little joke from the examiners there, and there's the H's. Name two stages of cellular respiration from which NAD may pick up hydrogen ions. Well, you've got glycolysis, link reaction, and then you've got Krebs cycle, and then away from that you've got oxidative phosphorylation. Then in all of these stages you're getting NAD turning into reduced NAD or NADH or NADH2. So we can mention any of those. So we could have glycolysis, we could have link reaction, or indeed we could have Krebs cycle. Hydrogen ions consist of one proton and one electron. So if we were to draw a hydrogen, it would be a nucleus with an electron going round. So the nucleus is H plus a proton, and around there we've got an electron E minus. So the hydrogen is a H plus and E minus. Explain how the components P and C operate to generate an electrochemical gradient. Well, P handily has got the right letter, so this pumps. If we draw it on this one, let's do it in a different colour. Pumps hydrogens into the intermembrane space by active transport. So pumps by active transport hydrogen ions or protons into the intermembrane space from the matrix. C, and these are proteins in the inner membrane. Well, these carry electrons. These are the electron transport chain. That's what they do. We don't want to say necessarily the full story for those, because the next question is what is the role of those. So these carry electrons, or you might say it's the electron transport chain. And then the next question is, well, what do these electrons do? And these power these pumps, these proton pumps. So these provide energy for the proton pumps. And naming the enzyme. So here's the enzyme that these protons, the H plus ions, are going to drop through down their concentration gradient, and we should be happy with the name there, ATP synthetase. That's the one that we've taught you to say. Technically people might give it other names like synthase or ATPase, but that's the synthetase is what our textbooks tell you to write, so I would go for that. Explain how this enzyme generates ATP. Well, we're saying is what's it going to make it from? ADP plus our inorganic phosphate going to ATP. How is it getting the energy to do that? We're going to talk about these protons moving down their concentration gradient. So using energy from protons or H plus moving down their electrochemical gradient. You could just say gradient there and you'd get away with it today. Electrochemical gradient. And we'd mention at this point chemiosmosis. There's a good thing to say there. So So pretty straightforward biology knowledge, no application for 14 marks.